Diponegoro University, or Undip for short, has one of the largest campuses in Indonesia and is located in Semarang, the capital city of Central Java. It is a place where students can develop their abilities and build their character and competence. Complete is a profile for our graduates, which stands for Communicator Profile. Undip has a Faculty of Science and Mathematics to accommodate the development of mathematics-based science and science in general. One of the flagship study programs is the biology. Undergraduate Study Program. In order to provide excellent service, the management leaders of the Faculty of Science and Mathematics and their staff actively coordinate and are fully devoted to manage every policy with full responsibility and professionalism.
Assalamualaikum warahmatullahi wabarakatuh. Very good morning, Excellencies, ASIN Assessor, Honorable, the Head of Biology and Chemistry Department. My name is Professor Dr. Widowati as a Dean of Science and Mathematics Faculty, Diponegoro University. On behalf of the Faculty of Science and Mathematics, I have the great pleasure to welcome the ASIN Assessor. The Faculty of Science and Mathematics has 6 departments and 11 study programs, consisting of undergraduate program, international undergraduate program, and also postgraduate program. As we know that there are about 4,952 students, including national and international students, and 180 lecturers. Faculty of Science and Mathematics has 17 professors who are from biology, chemistry, mathematics, physics, and statistics department. The vision of the Faculty of Science and Mathematics is to become an excellent research faculty with international reputation in the field of science and mathematics, as well as the development of its application. Synergetic cooperation both at the national and international level through collaborative and multidisciplinary research up to commercialization stage continues to be developed. As a research faculty with international reputation, of course, alumni of the Faculty of Science and Mathematics certainly highly competitive at the international level. The excellent and international reputation are the key achievement goal of the Faculty of Science and Mathematics vision. It means that the governance should be based on the international standard. As for one of the ways to achieve the key goal is through ASIN international accreditation of the study program with high quality standards. The process of international accreditation has been started with compiling the document regarding the teaching and learning process, research, community services, and the collaboration between the academia and industry partner, either from national or international level. The teaching and learning system has been carefully built to maintain its relevance with learning outcomes. I really appreciate the strong collaboration among task force team of biology, chemistry undergraduate program, and also the team of LP2MP Diponegoro University. Today, the ASIN assessment process is going to be assessor visiting for undergraduate program of biology and chemistry. Finally, I encourage all the task force team of biology and chemistry undergraduate program to fully observe at the assessor visiting assessment. I do hope the assessment will be run very well and successful. Thank you very much for your kind attention. Wassalamualaikum warahmatullahi wabarakatuh.
Good day. My name is Sabto P. Putro. I'm head of Biology Study Program. Biology Study Program is expected to have graduates which can work as researchers, teachers, entrepreneurs, bioindustrial workers, or environmental practitioners with the ability to utilize and develop tropical biodiversity in a sustainable manner. They are also have to able to produce high quality publication and technological innovation on the use of developments and biological resources. They are also able to carry out community service programs that educate local and communities on the utilization and development of the sustainable biological resources. Our study program consists of lecturers credited from various universities, both overseas and national institutions. Some of them are professors. The curriculum of biology study program has been prepared with reference to the vision, mission, goals, objectives, and learning outcomes of the graduates. Thank you.
Our biology lecturers, who are professional educators and scientists, are always active in carrying out their tasks and responsibilities. Our lecturers are responsible for fostering students so that they can achieve their graduate target according to the objectives of the biology study program. We understand that flexibility and open and clear communication between lecturers and students is key in achieving the expected graduate targets. Our students are supported step by step by the abundant availability of laboratory facilities as they develop themselves into prospective professionals and skilled scientists. The ease of access and convenience of studying and conducting research for students is a tangible form of the commitment of the biology study program to realize excellent service. At Undip, a laboratory is an academic facility for assisting students in becoming skilled and professional workers, thinkers, and scientists.
Oke, Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Good morning, everyone. I hope everyone is doing really well, even though we are currently facing a global pandemic, and everyone still stay healthy and safe. So, first of all, let me introduce myself. My name is Levina. As your MC today, I am an undergraduate student from Biology Department, Faculty of Science and Mathematics, Diponegoro University. First of all, I would like to say welcome and thank you very much for everyone's attendance today. And maybe without further ado, I would like to say a kind reminder here for everyone to maybe change your name, such with the format of your institute underscore your name. I'm sorry, maybe, okay, thank you for unmuting yourself. Okay, I will repeat the reminder for everyone to rechange your name for your as with the format as your institution underscore your name. You can see the example here. Mine is biology undip underscore and then Levina, which is my name. So while waiting for everyone rechanging the name, here I'm going to read to you today's agenda lists. So for the first session, which is going to be the opening session, followed by everyone listening to the March Song of Faculty of Science and Mathematics, Diponegoro University. And the second session will be the welcoming remarks delivered by the head of the Biology Department, which is Professor Sabto. And afterwards, we're going to proceed to the main event, which is the online course. The lecture series will be divided into two parts and will be moderated by Mr. Ruli Rahadian. So for the first part will be delivered by Professor Pieter Gell with the topic of biomonitoring of water quality. And the next part, the second part, is going to be delivered by Professor Retnanningsih with the topic of biomonitoring in Indonesia. So afterwards, there will be a discussion session. And lastly, we are going to have a closing session. So without further ado, maybe everyone could mute yourself because after this we're going to proceed to the next session, which is listening to the March Song of Faculty of Science and Mathematics, Diponegoro University.
Saintis harapan nusa bangsa dari penjuru Indonesia, menggapai ilmu alam semesta, tak mengenal kata putus asa, bersinergi dalam cita, kobarkan semangat dalam dada. Berkarya membangun alma mater tercinta, watak Diponegoro tercipta, kuatkan tekad di dalam jiwa, iman dan takwa sebagai bekal, amalkan ilmu ke seluruh dunia, bersinergi dalam cita, kobarkan semangat dalam dada. FSM terus berkarya membangun alma mater tercinta. FSM undi, maju dan jaya. FSM undi jaya.
Okay, now the song is already ended. We are going to proceed to the next session, which will be the welcome remarks from the head of Department of Biology for Mr. Sabto. Professor Sabto seems to be already here, and so the floor is yours, sir.
Yeah, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Very good morning for everyone. And we actually very happy for today. This is just the beginning of the year during the pandemic session. I don't know when it's going to be finished, the pandemic, but the show must go. The show must go on. Then this is the first series of a thematic lecture which is we have Professor Pieter Gell. Good morning, Professor Pieter. Thank you very much for this. I mean, you attend this meeting, and then also Professor Retnanningsih as the second lecturer for the speaker. Currently, we actually have established several research cluster in Diponegoro University, especially in biology department. The research cluster here related to the marine ecology, terrestrial ecology, biomonitoring, biomolecular, physiology, genetics, taxonomy, biosystematics. There is all of this, the biological science, the research of the biological science will be expected to establish more than two. Actually, we currently have established two cluster research, which are Center of Marine Ecology and Biomonitoring for Sustainable Aquaculture, and also Center of Paleolimnology, or CPALIM. For the next center, we expect to have for the next 5 years, we'll establish the Center of Tropical Biodiversity, Center of Plants Anatomy, Physiology, Center of Animal Anatomy and Physiology, Center of Biomolecular and Genetics. So I think this is the good time for the students. Currently attending this meeting is about 286. Thank you very much for the committee, especially Masuki and all the lecturer who are what we call advising the students to come over in this meeting. So thank you very much. And last but not least, thanks for Prof. Nanning. Without your effort, this might be not possible to invite a very special guest for today, Professor Pieter Gell, who are expert in about ecology, especially in paleoecology, Ramsar wetland. I don't know what is it, but I think it's talking about the ecology, right? And climate change. This could, cover many biological science and its application. So thank you very much for all. And back to you, Levina. Thank you very much. Hopefully the discussion will be fruitful, and we maybe got many information from the Professor Pieter Gell, and will inspire us to go further for the maybe research collaboration or etc. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Professor Sabto, for your heartwarming welcome remarks. Now we're going to proceed to the core event for today's online lecture series. But before that, I'm going to read to you maybe the curriculum vitae that Mr. Uli Rahadian has as your moderator today. So maybe some of you questions, who is Mr. Uli Rahadian as your moderator today? Now I'm going to read his curriculum vitae. He is a researcher who currently having a focuses in fields such as entomology, soil microanthropods, and also sustainable agriculture. And he's also a part of professional memberships in Entomological Society of Indonesia. And his educational backgrounds, first of all, he gained his bachelor degree in Bandung Institute of Technology in Biological Sciences in 1996. And followed the year 2000, he also gained his master degree in Bandung Institute of Technology in Biological Sciences. And in 2009, he gained his PhD degree in University of the Philippines, Los Banos in entomology. And for the experiences that Mr. Uli has, First of all, in 2011 until 2015, he is the secretary of Diponegoro University International Office. And in 2016, he is the head of Diponegoro University International Office. In 2017 until present, he is the chief editor in Niche Journal of Tropical Biology. And last but not least, in 2022, he is, until present time, he is the chief editor in Bioma Berkala Ilmiah Biologi. Now I've read you Mr. Ruli Rahadian's curriculum vitae. Now you also have the picturization of how credible he is as today's moderator. Without further ado, let's jump right in. And for Mr. Ruli Rahadian, the floor is yours, sir.
Okay, thank you, Levina, for introducing me. Yeah. Well, good morning, all participants. Good morning, students, and especially for Prof. Pieter Gell. Good afternoon. I think currently in Australia right now it's around 1 p.m. right? Okay. Firstly, I would like to read the curriculum vitae from first speaker, Pieter Gell. So he is a professor emeritus in Federation University Australia, and his educational background, he graduated from Monash University in 1981 for his bachelor degree, and then he continued to the same university in Monash University in 1988 for his master in environmental science, and then he got his PhD degree still in Monash University in 2007. And for working experiences, Prof. Pieter Gell, he started in Monash University in 1986 until 1989, and then he continued to Université de Paris in 1989 until 1992. And he continued and he moved to United Kingdom in Loughborough University in 1993 until 94. And then he came back to Australia to Adelaide University in 1995 until 2008. And then currently, since 2008 until right now, Pieter Gell is a professor emeritus in Federation University. For his research, of course, he do many research, mainly focuses on paleoecology and Ramsar wetlands, and he pay attention more on climate change issues, of course. Okay, we will begin for our lecture first from Prof. Pieter Gell. I would like to invite Prof. Pieter Gell. Time is floor.
Thank you very much for that
yours.
introduction, Ruli. I trust I can be heard.
Okay, thank you.
Thank you very much, and selamat pagi, everyone.
Selamat pagi, Prof.
Selamat pagi.
And it's selamat siang here in Ballarat. I live about 1 hour west of Melbourne, in southern Australia, where it's much cooler than Semarang.
Yes.
And we've had very wet 2 years following 10 years of drought. So we have a lot of climate variability here. Now I'll try to share my screen. Hopefully that is visible.
Yes.
Yes. Yes. I can
That's
see
the
your
full screen. Oop.
yeah, yes, PowerPoint.
Here we go. Yeah. Okay. Now I have a small laptop and a big screen in front of me, so sometimes you'll see me looking up, which is when I'm looking towards the big screen. I'll try and move that over there so I can see. Okay, so I think I was listed as the School of Science, Psychology and Sport, but as of last month, we have restructured Federation University and I'm now in what's called the Institute of Innovation, Science and Sustainability. Nevertheless, the topic of today is biomonitoring of water quality, and biomonitoring specifically is critically important because worldwide people are realising that it's not sufficient just to measure water chemistry to assess the impact of humans through pollution on waterways. It's critical to understand the impact of that changing chemistry on the living beings within the waterway. And people around the world are seeing that, so increasingly we're seeing means of monitoring the living, and the likes of Nanning and myself also work on paleoecology, which can look at bioindicators that are preserved in sediments, which give a temporal perspective to the monitoring process. Okay, let me go next. There we are. Okay. So freshwater biodiversity is critical to the world's biodiversity, and also through the provision of ecosystem services to humans. And we know that through provision of fish and fresh water, but also social and cultural amenity and the like. Yet freshwater only covers 6% of the Earth's surface, but hosts 9.5% of the Earth's animals. So it has a disproportionate level of biodiversity. The diversity of freshwater fish is similar to that in the oceans. Large pelagic areas of the oceans are large, have very low numbers of fish. Most fish are in the littoral or coastal zones. But critically important to understand is that while we might focus on marine resources and terrestrial resources such as rainforests in terms of biological conservation, the World Wild Fund for Nature have demonstrated that freshwater biodiversity is at greater risk than that on land or at sea. It has a negative 83% index, which is a comparison between biodiversity in 1970 and 2020. So it's declining rapidly, and it demands us to act very quickly. Also, the decline in freshwater vertebrates is considerable, and you can think of fish species, but also turtles in freshwater systems, as well as frogs and also freshwater mammals such as large river dolphins, many of which are at critical risk. And their risks to these systems are diverse, in wetland threats, but also from catchment processes. And while climate variability is important in impacting on numbers, human activities, particularly over the last 200 years, have been critical. David Dudgeon, many years ago, 2006, in fact, I think that is, you intend to look that up, identified 5 major threats to freshwater ecosystems: habitat degradation, the regulation of our waterways through dams and reservoirs, impact on water quality through pollution, exploitation, for example, consumption. In a very wet place like Semarang, that might not be all that obvious, but if you go to drier parts of the world, the use of river water as well as groundwater has substantial impact on freshwater systems. And in fact, places like China have lost over 30,000 rivers in the last 50 years, which seems remarkable to consider. Loss of species through bycatch, that is, those intending to catch large species tend to also in nets catch smaller species, and then the impact of invasive species, whether it be through the aquarium trade or through human-aided dispersal. These were identified as the major threats 15 years ago. More recently, we've published a list of emerging threats, and increasingly we're hearing of things like infectious disease, emerging contaminants, nanomaterials, microplastics and the like. But there's also increasing recognition of the impact of just light and noise associated with human activity on biota. You can imagine if you have 24-hour light, then species that move up and down or in and out or embark in drift down rivers lack those night-day triggers to drive those activities. And of course, changing climates is a critical one, and then the cumulative stresses which are exacerbated by climate changing and the impact of that on things like salinisation and calcium and the like. Harmful algal blooms are also emerging as critical emerging threats as well. So how has this happened? Of course, over 10,000 years since the development of agriculture, there have been measurable impacts of people on aquatic systems. As much as 7,000 years ago, we can see from the sediment record the impact of people on aquatic systems. But that has accelerated in the industrial phase with increased consumption of fish, crustaceans, shellfish, etc. And even more so since the 1950s, in the period that's now identified as the Great Acceleration. The consumption of water for food, fibre and energy, and all the embedded water in the food we eat, increased production of wastes, urbanisation, changing the catchment efficiency of systems, increasing and increasing the apex of big floods, and the empowerment of rivers, controlling floods, and the abstraction of water for irrigation and stock and human use. So these represent great challenges that are increasing or have increased over the last 50 years, and with human population will increase into the future. So we need to implement measures to keep pace with those changes, to mitigate the impacts on biodiversity, and to retain the ecosystem services that freshwater systems provide. So we need to become more efficient in water use, more innovative in how we treat volumes of water, particularly for increasing consumption. We need to reduce the volumes of water required for food production. And we need what's called integrated catchment management, which is managing all the aspects of the catchment to mitigate pollution, to try and improve water quality as a resource for ourselves and also for biodiversity. On top of that, we have ongoing climate change. In my part of the world, it will increase drying as well as temperatures. And we have to deal with sea level rise. Where you are, you have increase in intensity of cyclones and storms, and possibly increasing dryness in the dry season and general wetness in the wet season. The impact on wet systems might be increasing stratification, which might lead to increased nutrient release from sediments. Thermal extremes beyond the tolerance of some species, but higher temperatures advantage those blue-green algae, the cyanobacteria, that can be toxic. Increase in drying, and drying might lead to deflation of pollutants such as metals, raining down on human communities. We know there'll be changing rainfall patterns. Large-scale drivers of climate such as annular mode, the Indian Ocean dipole, El Nino sudden oscillation will change over space and time. It's likely to change the patterns of windiness, maybe less windy, maybe change the directions of winds. And the switching on and off of monsoons are implicated here. And orographic rainfall affects the direction of winds. Increased thermal energy, higher temperatures will lead to increased absolute humidity, which means that there'll be more water in storm, increasing runoff and flooding. And of course, rise is a major threat, with anything from 80 to 150 rise forecast as much as 15 years ago. And depending upon the mechanisms of sea loss and shedding large ice drifts from Greenland and the Antarctic, might be greater. Scarily, the last time the world was at 450 parts per million of carbon dioxide, sea level was 6 metres higher. So unless we embark upon low emissions programs, that's the forecast we're facing, but over several centuries. The impact of that on freshwater coastal marshes and mangroves can be modelled, but it's unsure. So let's look at aquatic and some definitions. I think we all know what pollution is. I was asked to define pollution when I was in grade 3 at school, so that's a long time ago, I assure you. Effectively, the direct or indirect impact on not just the chemical, but the physical, biological and radioactive properties of aquatic or marine environment, in such a way as to create a hazard or potential hazard to the health safety of any living species. So there's the importance there of biomonitoring, because the definition of pollution broadly requires it to impact or exist as a hazard to living things. The photo there is the impact of a copper mine in northern Australia. It was a heron that's died. It might not have died because of the high concentrations of copper, but one assumes that it might have been better without all the copper in the water. And pollution depends on the nature of the receiving waters. For example, if you add salt to seawater, there is little effect, provided there are no associated contaminants with that. Seawater is at 33,000 parts per million. It could probably go up to 34,000 or down to 32,000 or even a little bit more without it impacting particularly on the biota. However, if you add volumes of salt to pristine streams that have less than 1 milligram a litre, then you can have substantial and immediate impacts with small volumes of salt. So it very much depends upon the natural condition of the stream, and that's a river on the top there, Australia's highest peak, the Kosciuszko Mountains, and any delivery of salt into that system can have a substantial effect. In terms of mitigating pollution, it's very important to understand the source of that pollution. If you direct significant funding, but you haven't accurately identified where most of the pollution is coming from, you might waste a lot of money and have negligible benefit. So most pollution comes from direct or point sources such as outfalls. And that outfall there on the left is actually the main pipeline which takes water from the Murray River system to the city of Adelaide in South Australia. For many decades, they treated that water with chlorine to stop the fouling of the pipe so that the water flowed efficiently. But then they dumped the river water into the River Torrens, and of course the toxicity of the chlorine was sufficient to impact very heavily on the living things in the natural river. They eventually stopped doing that because of the impact of those pollutants on the biodiversity. But they were able to do that quite readily because the point source was very easily identified. You could see cause and effect fairly clearly in that photo. However, much pollution is from diffuse sources, and that can be wind-transported pollutants. In the big dust storms of Australia, many tonnes of dust is deposited in the Tasman Sea between Australia and New Zealand. Many, many tonnes. And that includes the salts and the nutrients that were on the surface of the Australian landscape. That can happen at smaller scales, but also bank erosion, overland flows can bring much less readily identified pollutants to a river or a estuarine or marine system. And in terms of phosphorus, because phosphorus is bound to clay particles, that diffuse source is a high proportion of the total, and therefore it's much harder to manage. Now, looking at some of the types of pollution, it's less obvious to understand that temperature can be a serious pollutant. These are two tropical species here, and it's possible in some reservoirs you can add warm water from cooling towers, from for example, and you create conditions suitable for tropical fish, for example, in temperate zones. And we have ponds in southern Australia where the temperature is only 10 degrees in winter, but the water is at 30 degrees, suitable for cichlids from South America and Africa. The bottom illustration is a picture of a piranha from South America. We don't want them in our waterways in Australia. They won't survive in 10 degrees, but they might survive in 30. But raising water temperatures can also affect breeding triggers. It can also reduce the amount of oxygen in the water and accelerate chemical processes, and these can all impact on the biota. Conversely, if you release cold water into warm water, you can also have a significant effect. While you might have more oxygen, cold water might suppress breeding of species that require warm temperatures to trigger breeding. And the Murray River is a good example of that, because the hyperlimnetic water, that is the water of the dam wall, which is cold because it's away from sunlight, can be released, and because of its high specific heat, it doesn't warm up for hundreds of kilometres, suppressing the areas that are suitable for fish to breed. And this is a worldwide problem in terms of the release of water from reservoirs, and our engineer colleagues are designing means by which to take water from the top of reservoirs to make sure it's suitable for fish breeding. So in rivers just to the east of me here in Victoria, there's been evidence of the impact of warm water releases on biodiversity. So monitoring programs and biomonitoring programs of sites with 50 species dropped down to 7 species. So that's 80% of species are lost on account of releasing warm water into a cool river. Climate change is also warming rivers, and it's already through monitoring been demonstrated that the rivers of Sydney are 1.5 to 2 degrees warmer than they were 40 years ago. And this will shift the type of species that exist in those rivers in the future. Salinity is also a pollutant, less easy to recognise in somewhere like Semarang, but of course, if you have land subsidence and rising seas, your water systems impacted by marine salinity. Salinity is measured in conductivity units as a proxy of parts per million of salt. Seawater is around about 50,000 microsiemen per millimetre or EC units, and salt concentrations are around about the equivalent of the Coorong system, or parts of the Coorong in the Murray River system are saline, but they are sometimes affected by the release of fresh water. And so a lack of salt can be a pollutant if the system is saline. The top illustration shows a freshwater lake in the inland of Australia filled by rising saline groundwaters on account of hydrological imbalance from vegetation clearance in the catchment. And because the groundwaters are now 20 metres closer to the surface than they used to be, there's little they can do to manage that wetland that switched very rapidly in the 1970s from fresh to salt. Salinity tolerance is an important parameter by which we would measure or monitor the impact of salinity. Salinity impacts upon organisms through their tolerance to the osmotic pressure of the salt. And if there are many salt-sensitive species in your waterway and the salinity increases, those sensitive taxa will be lost. They may well be replaced by more salt-tolerant species. And we can see, and I mentioned the Coorong Lagoon, a switch from relatively freshwater bird species to much more salt-tolerant bird species. And here is a picture of flamingos, which are salt-tolerant in southern France. And in freshwater lagoons, there'll be a different suite of species. So it's a salt tolerance or preference for that matter that can be used as a bioindicator of the salinity of a wetland. At around about 1000 DC, many species are tolerant to salinity, but as soon as it rises above 1000 and particularly above 5000, many species of organisms are impacted, particularly plants, and that leads to a loss of species diversity. The diatoms that Naning and I work with may shift from the bottom species there to the salt-tolerant one above it. That it doesn't mean that we lose species completely. There's quite often a of fresh preferring to salt preferring species. It's also important to recognise that salt, particularly sodium chloride salt, and we've all witnessed the school putting a piece of sodium into a beaker of water. Sodium is highly dispersive, and sodic-rich soils will disperse, break up, and be very easily eroded, whether it be through water or wind erosion. And that leads to the loss of fine sediments, and fine sediments carry phosphorus, and so salinity actually increase eutrophication as well. Another significant pollutant is acid rain, and it was poorly recognised for many decades, although perhaps to a degree it was resisted by industry, much like climate change has been resisted. But it was fairly well demonstrated now across Northern American and Northern European lakes that atmospheric fallout from sulfur dioxide, nitrous dioxides, which produce nitric and sulfuric acids, lead to substantial declines in the pH of receiving waters such as upland lakes. And that did impact significantly on particularly the fish biota, because, well, as soon as you exceed or drop below a pH of 5, the medium is released from solution and it can lock up into the gills of fish and suffocate them. And what was found in Scandinavian lakes was that the rainout of those acids led to a 100% loss of fish in many lakes. And it can also affect their reproductive biology by rendering many adult fish sterile as well. So there are massive fish kills in both Canadian and Scandinavian lakes, and they're now working to reverse that through liming and reducing the releases of sulfur and nitrous oxides into the atmosphere. Acidity can also be a factor in coastal zones, and now in fact in inland zones in the Murray-Darling and elsewhere, because throughout the last 7,000 years of stable sea levels, the seas have laid down sulfuric sediments. And where they're relatively stable under mangrove and salt marsh ecosystems, they lead to very little impact. But where there is marina and canal development at the coastal zone, they can be exposed, and those reduced sulfurous compounds can be oxidized into sulfuric acid. leading to acidification. And we can see substantial declines in acidity, in pH, leading to acidification of coastal systems, as well as inland systems in the Murray River system. We tend to ignore the impact of sediment itself, and the erosion, the agriculture, abutting riverways and lakes, the exposure to cattle and stock, and the direct waste into these systems can lead to an increase in movement of clay, colloids into waterways, and this can have direct impacts because many freshwater species, invertebrates, are filter feeders. And they not only use their gills for breathing, they use them for collecting food. So if you can imagine, if you put up a net, like a gill, like a net, into the stream, you start collecting not only food and oxygen, but sediments. And very quickly your gills are clogged up and you suffocate. So sediment pollution has the direct impact of eliminating filter feeding species in particular from polluted systems. So one of the best indicators of a sediment polluted system is the absence of filter feeding invertebrate species. Turbidity from this release of sediment also reduces light penetration. And so in this system, this is a Lachlan in Australia, the sediment and the turbidity reduces the ability of the light to penetrate to the bottom of the river. And so aquatic plants are not able to grow. We know that once the water was clean, rich in aquatic plants, and now they are lost. And so the habitat for fish in the Retnanningsih system has been lost as early as the mid-1800s, in fact, but certainly in the last 50 years. So to monitoring, how do we monitor these things over time? Well, if you wanted to monitor something, the best time to start was many decades ago. But unfortunately, we can't do that retrospectively. We're not monitoring a stream. The best time to start is today, because we don't know when the impacts are going to happen, and we can get the benefit of long-term pre-impact data if we start monitoring as soon as possible. So freshwater scientists around the world advocate for government to invest in monitoring programs. And of course, well, they would say that, wouldn't they? They want more money for research. But without that long-term monitoring, you don't know how your system is changing and responding to the impacts of people. There are many ways to go about it. Multi-probe data loggers, they're relatively expensive, but they're efficient in that they don't use a lot of human time. Archive data. Over the many years, people have taken and stored away data sets from rivers and lakes that can be accessed, historical data. Biomonitoring that can start today. And one of the indicators that I use are algal diatoms. And there are indices, because they're international, there are indices we can use for salinity, pH, and nutrients that are available from all around the world and that are now being developed by Diponegoro University in part of the world. Very easily sampled, and these large data sets can be used to infer water chemistry change. There are many bioindicators. Indicator is diverse, abundant, and so easily sampled, relevant and easy to identify, then it's a very good indicator. By and large, that means they have to be small. If they're large and easy to move, such as these water birds, they're usually less responsive to the water. So the best indicators are often the common, diverse, small indicators, such as algae and invertebrates, and less so the large ones, such as mammals, birds, or reptiles. Even so, nowadays, there's a lot of investment in environmental DNA, where a single water sample be used using DNA technology to identify the presence and absence, and sometimes abundance, of all the things that have been in that waterway in recent times. So, other pollutants include organic pollution, and we know the release of nitrates and phosphates will drive, or dissolved organic carbon can drive productivity, which is respired by decomposers and can lead to the loss of oxygen in water bodies. This is widespread where there are any humans. We now have extensive anoxic zones in our oceans, and it's been demonstrated through sediment records that most of the large deep lakes in Europe became anoxic, at least in their bottom, over 100 years ago. So industrial development very soon had an impact on large lakes, such as Lake Geneva in that top example there, where they're having fish kills today. Nitrate and phosphate is essential for algal growth, as is phosphorus, not only as a fertilizer, but also a natural phosphorus bound to clays. This leads to eutrophication, and the algal material can die and again be metabolized and lead to anoxia. And the example in the top right there is a source of phosphate, because in Brazil, they wash their car in the river, or at least they did in this instance, and they use detergent, which is high in phosphorus. So it's not just phosphorus through drainage systems. In this case, it's direct use of phosphorus in the river to wash their combi vehicle. Nutrient monitoring can be used chemically, just with various test kits. You can measure dissolved oxygen as a proxy for eutrophication. Spectrophotometers can be used in the laboratory, or if you have funds, you can use external analyses from accredited laboratories, but that can be quite expensive. We need nowadays, as I mentioned earlier, to biomonitor the impact of nutrients. And again, I suggest that one of the best indicators for nutrients are diatoms. There's international indices such as the Diapo index in Japan, Omnidia, which is an international index, there's a Dutch index, there's an Australian index that can be used quite readily in Southeast Asia. Another proxy is chlorophyll A, which is readily measured as well. And here we have a program in China where they're monitoring the largest blue-green algae bloom in the world at a lake just downstream from Nanjing. And you can see that Taihu, and you can see there's bloom as far as the eye can see here. Heavy metals are also important pollutants, and they can bioaccumulate and biomagnify through the system, interfere with normal bodily function, interfere with not only humans, but right the way through the food chain, and even single-celled algae can be affected by heavy metals. And the old Mad Hatter's story is associated with the release of mercury from the use of mercury in the production of felt hats. But the Minamata disease was also known from Japan. We have in Australia mercury released to extract gold. So we have major problems with mercury in our food webs in terms of fish production. So monitoring of heavy metals, we can analyse sediments using various chemical analyses, XRF or ICP. Again, archive data, there's worldwide sampling of heavy metals. Again, that requires funded analysis. I just wanted to shift to talk a little bit more about lakes. Because rivers flow, they will change in space and time. Lakes also change in space and time on account of depth zonation. So they exist as a gradient from shallow water near the edge where there's light, out into the deeper waters where there's deep, without much light. And so aquatic plants tend to follow those depth zonations. And so that also drives a variable tolerance to inundation and desiccation. You live in the margins, and lake levels vary. You have to be tolerant of drying out. Those less tolerant of drying out generally live in deeper waters. Now, water depth is critical for driving biodiversity, as exemplified by the really high diversity of those Rift Valley lakes that are a kilometre or more deep in Africa. Most importantly in lakes is the issue of stratification. Deeper lakes, such as those I mentioned in Europe, have an upper water body and a lower water body and a cline which prohibits the mixing of the two. So they're effectively two lakes in one. The upper water body is warmer because it's receiving light. It can be fresher because it's receiving rainfall, and it can have a different chemistry as well. The gradient between the two might be quite steep, as you can see in the right-hand side of that diagram. And if you've been in lakes and swum, you can feel the abrupt change in temperature. This drives a chemocline where the less dense water, which is fresher, sits over the saline denser water below. Quite often, that chemocline can have oxygenated surface water and deoxygenated lower water. And in fact, the whole productive system might only exist in the top 5 or 8 metres of the lake because of the lack of oxygen at depth. Now, this layering or stratification of lakes can vary greatly through pollution, through temperature changes, changes in mixing from wind regimes, and so that can drive huge changes in lake productivity. Annual circulation patterns. In the tropics, for example, you often get mixing overnight, what are called polymictic lakes. But in some places, you only get mixing when the lake cools down, which breaks down the stratification, the temperature stratification, to allow mixing of those lakes. And you might get the oxygenated layer mixing with the less oxygenated layer. And of course, so that's a seasonal thermal variation driving mixing, but that could also be affected by the prevailing winds, which might have episodic mixing. But as you can imagine, if you have a huge mixing event, then you might get huge fish kills because you're mixing the section of water with oxygen with huge volumes that are without oxygen. And so the fish might be suffocated on account of a lack of oxygen. So we can't always attribute fish deaths merely to pollution, whether it be heavy metals or the like. It might also be on account of the changing layering or stratification of the lake. Turbidity is important in that stratification because it impacts upon light. And we can see here in yellow the deepest plants. And if you have a high Secchi disk, that is high light water clarity, and 40 metres, you can have plants growing up to 140 metres deep in a lake. So you can have a very productive system. But if turbidity means that your light penetration is only 4 or 5 metres, then plants can only grow to 15 metres. And even worse, if your turbidity means that light only penetrates the top 1 metre of the lake, then the plants are confined to the top 1 or 2 metres where they can photosynthesize. So turbidity can impact the productivity of a lake enormously on account of this light-plant relationship. It's important to understand not only sources of sediments, but sources of nutrients, as they drive the metabolism of a lake and the biofilms, which are an important basis for the food web. nitrogen and phosphorus are also important components. Phosphorus sometimes might be limiting because it can be readily consumed, but in anoxic situations, as I've discussed in these deep water lakes, the phosphorus can be remobilized and become more bioavailable with anoxia at depth. So the stratification, the layering of the lake, can actually increase the release of nutrients and can drive lake productivity. And if there are large volumes of nutrients, that can lead to massive increases in hazardous algal blooms, such as cyanobacteria. So understanding whether it is catchment source, local source, or internal source from sediments is important in understanding how the food web functions, how you mitigate the impact of elevated nutrients. So in terms of mitigating nutrients, if you have had many years of nutrient release, and there's another example from Brazil in the top there, it's very difficult to control phosphorus levels because there's so much phosphorus in the sediments under the lake. Best means of managing that is actually to aerate the lake, excuse me, to reduce the incidence of cyanobacterial blooms. So in terms of understanding lake change, it's important to establish chemical and biological monitoring because we need to see the biological response to chemical change. We need to start monitoring now if we haven't already done so. We can also access archived data. Important also increasing photographs. How many macrophytes were growing on this 40 years ago when somebody took a photo with their family, for example, is good archival data. Paleolimnology provides continuous sediment records of particularly micro-indicators of change. And we can also increasingly use remote sensing of data to look at lake levels, lake condition, lake vegetation, and increasingly lake water quality. And I'd encourage anybody to undertake lake water projects in any of these sorts of approaches. To do all of these at once would be very challenging, but certainly you can contribute to larger projects by embarking upon one or several of these different approaches. Paleolimnology is about coring sediments. And here you can see on the right, you can take a continuous record of sediment, sometimes for 100 years, sometimes for 5,000 years. It provides a really long-term baseline against which to compare impacts over the last 200. You need technologies to date those sediments. You can apply chemical analyses to the sediments to look at chemical change over time. And buried within those sediments are a range of biological analyses, and the technologies are becoming more diverse as people investigate new ways of looking at the biological remains that are buried away with sediments over long periods of time. I also wanted to mention the importance of the catchment. And in urban catchments such as in Java, it's critical to understand the impact of impervious surfaces, roads, concrete, house roofs, on the changed hydrograph. And you can see here an unurbanised catchment here has a 1 cubic metre per second discharge every 3 years. But you find every 3 years, it's 3 metres per second in a impervious catchment. 6-fold increase in incidence of those sorts of flows. And so increased flood threat can lead also to warmer runoff, but also increased erosion, increased provision of nutrients, increased provision of sediments to these systems. So the management of nutrients is not only about direct sources, but it's more integrated catchment management, managing both pollutants, but also flow from your catchments and the impact of diversions for domestic use. And you can use archived hydrographic and climate data to try and marry up nutrient load data with your flow data to better understand the system. So I'll finish there, recognising that climate change is upon us. This is the largest lake in New South Wales. It's a large lake today, but when I took that photograph, it was a mere puddle on account of the drought we had. It's generally about 30 kilometres by 50 kilometres lake. So we're going to experience increasing climate variability, but even so, the last 200 years of human impact on our systems mean we need to recognise the impact on aquatic systems, measure the impact through monitoring, and invest in water research into the future, because it's one of the most critical services that our natural systems provide people, and we need to understand how it's changing to ensure that service continues. So thank you everybody for listening, and I think we're having questions after the next presentation, although I'm happy to take one if there's change over. So thank you very much.
Okay, thank you very much, Prof. Pieter, for your very informative lecture right now. For us and question session, please be patient for all, because we will conduct after second presentation. Okay. But if you want to ask question that right now, you may type, using chat facilities. Thank you. And then for the second lecture will be delivered by Prof. Retnanningsih Suprabhawati. Firstly, I would like to read the curriculum vitae from Prof. Prof. Retnanningsih, also known as Prof. Nanning. Okay. Okay. Our second presenter or speaker or lecturer is Professor Dr. Retnanningsih Suprabhawati. So her educational background, he got a bachelor degree from Gajah Mada University, and then he continued for his master and Master of Applied Sciences, especially in environmental science, and I think this is Western Sydney University, right? Australia. And then he got his PhD degree in environmental science in Gajah Mada University. And for Prof. Nanning experience, we can see here many experience. Currently, she is a Vice Dean for Resource and Innovation, School of Postgraduate Studies in Diponegoro University. And then she is also head of Senate Faculty of Science and Mathematics until right now. And then he was a head master program biology and from 2013 until 2017. And then she was a secretary master program biology also, started from 2010 until 2013. And then she was the head of department biology from 2003 until 2007. Prof. Nanning already published 55 publications until right now, and then her area of interest, especially in water equality and also microalgae as bioindicator and FICO remediators. And yeah, for activities, many activities here, including some of them is expert for Ministry of Environment and Forestry since 2011, and then he conducted research collaboration with Federation University Australia since 2009, and then he She conducted research collaboration with ANSTO, Australian Nuclear for Science and Technology Organization, and I think many more. Yeah? You can read here. Okay. So the topic for lecture from Prof. Retnanningsih will be biomonitoring in Indonesia. Okay, without further more, Prof. Nanning, the time is yours.
Okay, thank you, Pak Ruli. Okay, let me share my PPT first. Okay, can you see my PPT?
Yes, we can see your PowerPoint.
Okay, okay. Thank you very much, Peter. I would like to inform you that the audience for today's class, we have a student, not only undergrad student of biology, but also master students of biology, and then environmental science, as well as a PhD student. And also there are many colleagues from another university that join with us. As I know, there is Agatha, Cecilia, and Diana from Unsud. And then Pak Mislan, thank you Pak Mislan, and then as well from Udinus, and also the colleagues from limnology and PPDAS, Hutan Lindung, kemudian PRP, SDI, and I'm sorry that I cannot say all of you together, but I'm happy that Yuli able to join with us also, because I remember whenever we are coring at Lutan Lake. Okay, that is my own outline. So I will be more theoretically, and then Prof. Pieter already mentions about the how monitoring program in Australia with their database and what happened in Indonesia. Okay, so whenever we are doing some activities in the monitoring activities, usually it is physicochemicals as well as chemicals, but once it is sometimes it is very expensive, and then sometimes it is cannot detect the biochemical changes over a long period time. So just reflected the water quality, for example, at the time when we collect the samples. So that is why it is then developed the using the organisms that able to be applied in the monitoring activities. So what is the biomonitoring? Okay, so the biomonitoring is the systematic measurements of compounds or detections of a cell or even cell molecules alterations in the living organisms with the purpose to identifying, assessing potential hazardous exposure. So you already have the examples how the biomonitoring nutrient, heavy metals in Australia, and see that in biomonitoring, it is consists of recording species diversity and abundance across different locations and times using a range of ecological census technique and taxonomic identifications. And whenever we use this, it seems later on we will try to discuss about the conventional biomonitoring and then the next generations of biomonitoring. Okay, so if we try to have a look this data, this is the studies that the length of studies is less than 1 year. It is very high. And then if we have a look in the 10 data, it is already just like this one. But if we have long-term data, the data will be very fluctuated. So it means that whenever we only have a small data, for example, for 10 years, for example, sometimes it cannot reflect the actual conditions. Okay, so there are differences between the biomonitoring, biomonitor and bioindicator. So biomonitor is an organism or part of organisms or in the community level that contains information on the quantitative aspect of the quality of the environment. So we are talking about the quantitative. So we are talking about the data, quantitative data. So a biomonitor is always a bioindicator as well, but a bioindicator does not necessarily meet the requirement for biomonitor. So usually for biomonitor, if passive, the plants, for example, that naturally grows in the study area, but actively we might be able to place the test plants in the study area. So if we are talking about biomonitors, usually it is related with the bioaccumulations. So what is about the bioindicator? There are compliance indicator and then diagnostic indicator and early warning indicator. So I just want to focus on the early warning indicator that usually will be give early warning systems like what happened in the next time. And the bioindicator have to fulfill the requirements. For example, they have a potential of good indicator ability, and then response reflects the whole populations, and then measurable responses, and usually the species of or group of species have to be abundant and common. For adequate of local population density, and then common means that is sometimes it is cosmopolite, and then a relative stable despite moderate climates and environmental variability. And then it has to be already well studied, ecology and life history, and then taxonomy, easy and cheap to survey, as well as economic and commercially important. And this is what happened if we have a look like this. So it means that what happened in our ocean, in our sea, something have to be happen in the deep sea there, or in the terrestrial ecosystems. Whenever these species go downward from the mountains, for example, it should be something happen in that mountain. So that is the concept of using the organism as a bioindicator. So this is another illustration that whenever the environments in the for example different in the temperature and then water velocity, food, so it will be influence in the species level, for this one species level, and then also will influence in the populations level, and in this is also influence the growth in the community level. So whenever we are talking about biomonitoring, we might be able in the species, population, as well as in the community levels. So the bioindicator is already mentioned before that it helpful, objective, and straightforward and reproducible, and They are usually can be utilized in the various from the cell into the higher environments. This is the illustrations on how using the plant indicator and then animals indicator as well as microbes indicators, or using the organisms. We call it bioindicator. So biomonitoring, if we are talking about the quantitative data, bioindicator, if we are talking sometimes it is qualitative expresses by the organisms. So the bioindicator can be divided into the pollution bioindicator that detect the presence of pollutions, and then ecological bioindicator that detects the changes in natural and surrounding and their impacts, and then the biodiversity indicator that detects in the biodiversity and the species present, and then environmental bioindicator that detects or monitor changes in the environment. So we are here. Okay, that is what happened whenever there are something happen in our lakes, for example. So we might be able to use kinds of phytoplankton as well as zooplanktons that already studied very often, and then the reference that able to be used is also very high. And then the other thing is about the biomarker, that the function measures of exposure that is characterized at the suborganism level of biological organizations. So when we are talking about bioindicator, it means that is in the individual populations community. But whenever we are talking biomarker, it's supposed to be in the histopathologies or biochemicals or in the molecular or cellular levels. So this is the difference between the biomarker and bioindicators, that biomarker is in the subcellular or cellular, and then the bioindicator through individual until the communities. So it depends on what our target. If we would like to have a look the ecological relevance, so we have to use the bioindicator. But if we would like to have the sensitivity of the stressor, so we have to use the biomarker. Okay, so let me started bioindicator in the species levels. If we have a look this birds, the crows atau burung gagak, in Indonesia we call it burung gagak. We have at least 8 species of crows that under at least IUCN, and all the crows indicated, and people, even our ancients, had believed that crows related with the dead. Okay, so that is it means that our ancients already know about the concept of bioindicator. They use the crows as indications that somebody dead in their village, for example. And also see that the fish is the good bioindicator. See, a few weeks before, even for more than 250 kilometers. But if we use, for example, chicken, for example, it is only 20 kilometers and just 1 or 2 minutes. So the earthquakes already come up. And I will show you about this is the endemic fish, Java medaka, that have the potential able to be used as a bioindicator, because Orisias javanicas have able to be found in the freshwater as well as in the marine water, and then see that is its sensitivity to the heavy metals, and then also this medaka is also found in Semarang. That is why I choose this, and then also able to be found in Maluku, in Sulawesi, Kalimantan, and probably in another part of Indonesia as well. And why we use the indicator species? Because whenever there is a problems in the catchment area, and then they will be come up to the water ecosystems, and then the indicator species, for example, we use the meflinim, and it is in the low stream. But if we found tupifex, cacing merah, it means that the pollution level is high. So we use the species levels in that area. And as well as whenever we use the diatoms, this is the diatoms indicator of pH, list of diatoms. Actually, it is more than that. And some of those diatoms are found in Dieng, in Telaga Warna, and it is important that whenever we have the fossils of diatoms, we might be able to reconstruct past conditions in that lake. So in the species level, we might know that see that the Aulacoseria ambigua dominated in the upper layer. It means that in the upper layers from 1 until 10 centimeters, it is in the high levels nutrient, and then but in the lower layers, whenever we have a look through Aulacoseria ambigua, it is less concentration nutrient if compared to the upper layers. How about the populations or community levels? So whenever we have a pollutant here, for example, heavy metals, and then it have to be influenced the biotic factor, the organisms, and then the abiotic, for example, temperature, precipitations, as well as nitrogen part, and by the process it will be able to express by the species there or group of species that be able to be found in that areas. That is the simple to make it understanding that if the nutrient input in this lake, the alga bloom will be happen, and then the oxygen will be depleted, and sometimes the fish will be died. Why depleted? Because there are the decomposer or decomposition processes, and the plant also will be grows very well. It is whenever the nutrient is come up to the lake. But what happened is whenever the nutrient in the excess or in the high concentrations, the fish will begin to die, and then the increase of growth alga. So the concept of biomonitoring is whenever we have this data, we might be able to know about what happened in the catchment area, what happened with the nutrient, for examples. So in the populations and community levels, the decrease of abundance and diversity to the emergence of morphological deformity, and sometimes they have accumulative impacts. And the bioindicators do not respond to all disturbances and are affected by seasonal variations. But biomonitoring indices are developed for specific regional by local biotic assemblages that shows regional variation based on the organism sensitivity to tolerance. So the applying of these indices to the other region might be affect its capability, functions, and reliability. And this is the data fresh from the oven that actually that is Jasir project. My student, that when we try to put the keyword of bioindicator, water quality, Indonesia, and then it will be found more than thousands and 400s, almost 1,500s. But if we try to break down, and there are only not too much that related in the international journals, and also related that when we are talking about bioindicator, we are talking about water quality, and then bioaccumulations, and then in this part also talking about diversity. So this is the bibliometric that very easy to done using Viewer. And then if we are just focus in Java, so we found almost 300 publications, and then also against related the water quality, also related with monitoring activity, and then another things like some kinds of heavy metal, salinity, and others. And this is also, this is in Java. So based on this data, seems that Jasir found that if we are talking about the water quality and then river pollution, groundwater, it will become the data like this one. And there are 10 increased data by the time. But unfortunately, that internationally, mostly it is published in the conference proceeding. And this is until this one, it is this is journals, and then this one also a journal, but mostly in the proceedings. And this is the examples that I choose from Musi rivers, that we are also in the community level using subtropic index, and then based on the subtropic index, it can be said that the Musi River stay still in the good, relatively good and moderate conditions. And this is also another examples how to use the macroinvertebrates in Muara Jaya, and then see that the EPT and then non-EPT. The EPT means that by these higher concentrations mean that in the Muara Jaya is in the good conditions whenever compared to the another sites. And when we try to compare, there is a publications that try to compare how to use the diatoms and then macroinvertebrates and then fish. So the diatoms are not good for indicator in routine monitoring programs in the Wei River, but macroinvertebrate, it is good for indicating nutrient level, and the fish is a good indicator of spatial changes. Again, that it is depend on the location. Sometimes whenever we try to put together and use together, probably we have a different results by these organisms. And then in Indonesia, the foraminifers is also already used as a bioindicator of coral reef. Actually, in the beginning of 2000s, I used foraminifers as a bioindicator of water quality as well. And there are index of foraminifers that also able to talking about the conditions of coral reef using this index, as well as the functional group of species foraminifers that found in the coral reef ecosystems. And then another things that we might be able to use is diatom index. So that is just one examples of the formula. Actually, there are omnidia that Pieter also already mentions about omnidia. So we might be able to use omnidia to calculate a diatom index. So okay, so I will try to show you how we try to use the diatoms index for Telaga Balikambang. So we already take samples from Balikambang. So this is the water that able to be seen, but actually the green color, it is also part of the lake, because underneath it is see that the Balikambang lake, it is like what should like jar, like that one. So this is part of the lake, and the problem is land use changes, and then pumping the water for irrigations, and then reduce water quality, and dry it in the dry season, and then in some part somewhere here it's already changes into the agricultural land. And this is the Shannon-Wiener index that whenever we try to compare four sites from Balikambang, that based on the Shannon-Wiener index, it is in the good conditions, and usually also whenever we try to compare the evenness and dominance index, it is also in the moderate conditions. But whenever in the species level, we have a look at Nitsia and then Conphonema, Selapura, Pupulum have the highest portions and then dominated in the Balikambang. And when we try to calculate the diatom index, it can be seen that based on the percentage that IBD and then IPS and IDG, it is although it is these indexes are developed in mostly in the Europe, but it able to be applied in Indonesia, and then provide the data like this one. See that when we have a look the diatom index from the Balikambang Lake, so based on the IBD, IDG, the Balikambang in the good or in the oligomesotrophic conditions, and then and also I will try to compare with another result from Chepong Lake. It is also in the Dieng, and if we compare with this data, it seems that they also have similar results. And this is the Balikambang, the what should I call the eye tracks, like the scan of sediments, and then in this level 100 between 80 and 120 centimeters, it is recorded that is the period of frequent flooding. So that is another indicator whenever we are trying to put together all of things. But we haven't finished to identify the diatoms yet. So this is the data that we already have. And this is also in Cepong Lake with the similar problems that pumping water irrigations and then land use change for potatoes plantations. And this is what happened whenever we try to make a diagram of the richness of taxa. So in the lower layer, the number of taxa is higher if compared to the upper layer, as well as the Schneider-Wiener diversity data that in the lower layers is higher when compared to the upper layer. But the lower Schneider-Wiener in relations with the higher, the less of dominance index. And this is the evenness index, and the results is a bit different because in Cepong, Fragillaria dominated if compared with Nitsia, for examples. And this is the diatoms index that we have. So the IBD and then IPS indicated that Cepong Lake is in the moderate conditions, why the IBD and then IPS and IDG represents more than 70% of the species. It means that almost 30% of IBD, the species that were found in Cepong, do not available in the database of IBD as well as in the IPS. IDK, why it is 100%? Because it is in the genus level. So it is 100%. So this is another examples of monitoring. So the pH in the Warna is acid, and then in Pengilon it is fluctuated from acid into alkaline. Why? Because there is a seal in here. In the wet season, the water from Pengilon will be drained to Warna, but in the dry season, the Warna, because pumping activity is very high in here, so Warna will come up here. So in the dry seasons, the conditions like this one, and we already collect the sediment samples in here with Pieter. We have 210 centimeter sediment, and it is sedimented more than 13,000 pp's, and this is about 135 centimeters. It is sedimented more than 8,000 pp's, and in the 25 centimeters, it is sedimented about 125 years. And this is also the conditions in the Telaga Warna, because we only have 150 centimeter sediment cores. So what I have discussed is about using biomonitoring with the conventional activities, means that we collect the sediment samples and then preparations of diatom, for examples, identified, and then put the data. The next generation is using the eDNA for biomonitoring activities. Why? Because this is the conventional bioindicator, and then we try to rebuild the new bioindicator diatom apple to be used. And we already started this year to analyze, to do the eDNA using the samples from Balekambang. And the good news is we have success for the extraction process, because we have to trial and error. We have success, but at the time we still waiting the equipment to do more activities until come up to the name of species. So the EDNA piece is how to use this is the ecosystem interpretations, and then try to use the extractions and then the primer that might be able to be used. We already come up to this states, and then at the moment we're still waiting for this step, and then hopefully the data based on the eDNA will be in line with the data conventionally, and the eDNA and meta-barcoding is already used in the terrestrial as well as in the estuarine, and then collect the sediment samples or fresh water, and this is a marine and then fresh water, all of things that we might be able to identify the organisms in the sediment samples. And this is the the result from the previous publications, that from the if compared with the conventional and then meta-barcoding, it is similar. Even in the conventional, probably it is still a lot of database, but in the meta-barcoding, sometimes the problem is in the database. Okay, so but it's still similar activities, and this is another things that whenever using the diatoms and then cyanobacteria and then cladocera, and then put together how they able to, whenever we put together the reconstructions of what happened in the catchment area, will be very good expressed for the long periods based on the diatoms, the littoral community like this one, and then in the deep part, and then the cladocera also have a good potentials. How to use the biomonitoring activities related to the environmental management. So human activities will produce the pollutant and then release pollutant into the environment, and then will have the long-term impact of pollutions. And the strategy that able to be developed have to be based on the biomonitoring activities. So the restoration program that able to be developed is in the sense of economic, social, and environmental friendly. So the biomonitoring for water management that is that bioindicator can be employed at a range of scale from cellular to ecosystem levels, and then they bring together informations from biological, physical, and chemical components. And then from the management perspective, bioindicators inform us action as to what is and is not biologically sustainable. Okay. So that is all that I can share. Thank you. So I deliver to Pak Ruli again. Thank you.
Well, thank you very much, Prof. Nanning. It is very integrative lecture today. Yeah, and then it's time for us. Yeah, invite participants if you have any question. And I read in chat facilities here, some participant already give the question, and then Prof. Pieter already answered for some also. But I invite also participants who want to ask question directly. Please raise your hand, yeah, through the emoticon raise hand. First, while I'm waiting for the other question, I invite here there is the watershed management and also Abdul Qadir. If you want to ask directly, or might be you need further detailed information, might be, yeah. For example, here from, yeah, we don't have any name here for watershed management, already answered by Prof. Pieter, but he asked again with please explain more about dry biofilm. Probably Prof. Pieter, you can answer directly through this if you want to explain more about this.
Yes, I have just put up a post. But, if you look at some dry rivers, you can see mats of dry algae around the sides. There is a prospect that algae, if analysed, would contain the bioindicators or remnants of those bioindicators that reflect the condition of the water when it was last flowing. It's likely that if the indicators from the time it flowed before that were washed away, so it should reflect past river flow. But again, that's making something out of nothing, because when a river is dry, there's not much bioindicators living.
Oh, okay. So there is another question here for Prof. Pieter and Prof. Nanning. Yeah, I will read this one. Glad to be part of the session. I have some question about the water quality. Is the assessment of water quality with chemical, physical parameters not been effective in providing protection and control of damage to water ecosystems? And otherwise, is the water quality assessment with biological parameter has been more effective? This is from Ilham Yasir. And then what is the solution for developing the right water quality management? Thank you.
So I'm happy to start there. So that goes to the question of whether the release of, for example, salt water into a river is a pollutant. If the river is already slightly salty, then it might be a natural release. But if you know that this river had assemblage of biota and some of those have disappeared, then you know you've had an impact on the ecosystem. It also goes to increasing the relevance of the monitoring, because we're not all that concerned in the world about the actual chemistry of the water. It's the impact of that chemistry on the living things, most important, even though we know it's the water chemistry that is the driver. So it's about looking at the impact, the biological impact, and not the stressor or the driver of the change in the system. That said, it's very important, we know, to measure both the chemistry and the biology, because once you calibrate the biology to the chemistry, you can much better understand how the system is functioning and what is the causes of the change. So Nanning and I know that if you switch to things like Retnanningsih palea in the diatom community, then you have nutrient enrichment. But if it's amphora coffeeformis, then we know we have a big increase in salinity. So by looking at the chemistry and the biology, you become much more clever in being able to interpret change from just the living things.
Okay, thank you. Might be Prof. Nanning, you will add some more information?
Yes, okay, thank you for the question. So I just try to edit that. Yes, indeed, that whenever there is a problem in Indonesia, in the dry seasons, the aquatic ecosystems almost all dry. For example, in Rawapening, whenever in the dry seasons, it is almost 2 kilometers dry. And then more than almost 2 meters water volume reduce. And in that part, we try to collect the samples, and then because once it is occurs in the what I mean says in the wet seasons, in that area it is covered by the water, but in the dry seasons it is no water at all. And then we try to collect the sediment samples, or to collect the, what should I call, the plants, aquatic plants, stem or root of plants, and then try to analyze, and we have the data. And at least by the identifications of the species, we might be able to know the conditions based on the characteristic of the species. Because whenever we are working with diatoms, so we might be able to know if, for example, Cyanidra dominated, it means that the organic is probably it is high, and it is always remain like that one. And then whenever we try, we found Eunotia, so the pH usually is very low, kinds like that one. In Indonesia, we have the new government regulations last year, and unfortunately there are only physical and chemical, but good that they already separated the river water with the lake. And previously, river and lake just put together about the concentrations, the maximum concentrations for water quality standard. But the newest one, it is separated river and lake. So the good things is whenever we have a data, chemical, physical, as well as biological data, it will be very support each other. When we already have the biological data, so we might be able to explain the conditions, and then compare with the physical and chemical data that we have. Of course, it is in the upper sediment, or in the water quality, and then by this way, I think it will be see that we might also be able to analyze which parameter that have influence deeply in such organisms, and then how about the other organisms, usually also different. That is all that I can edit, Pak Ruli.
Okay, thank you, Bu Nanning. So I see time is almost almost 12, but I still give opportunity, one question. If you have one question directly, from students, might be just directly raise hand and then just say your question here. Before we end our lecture series. Okay. There is a question here in chat room. Question for Prof. Nanning. Okay.
Okay, thank you, Cecilia. This is our alumni from Magister Biology, and now she is a lecturer in Unsud. Okay, so if the lake is already dry, try to collect the sediment samples. That is what we try to collect it from Balekambang. We just we able to collect more than 400 centimeter sediment core from Balekambang. And then from Pengilon, we have to use the boat and then try to collect the sediment samples, and then we collect it 210, and it is easier whenever. Thank you, Peter, for the Russian corer. It is easier than piston corer, and then I already have more than 400 centimeters from Balikambang, and it is not in the water bodies, but in the edge of the lake, and it is easier, and the layer is very good. So that is what you have to do, Rani, to collect. It is easier to collect if it's already dried out. That is what we tried to collect from Rawapening 2 years ago. Whenever it is more than 2 kilometers dried, and then we try to collect sediment sample from there. Okay, thank you.
Thank you very much, Prof.
Yeah.
Okay. Okay. Now it's already 12 p.m. Then I think,
Ruli,
yep.
kayaknya ada yang raise hand.
Oh, raise hand. Oh, okay. From Undip, Miss Elita. Okay.
Thank you for the opportunities. Good afternoon. This is Naning. Basically, my background study is not in biology science, but I have a question. How we determine in a water body in the bioindicator sampling? Is this like to count the population density, or we can take the sampling in a certain point?
Okay, is that?
Yes, that's all.
So that you means that how to collect the sediment samples or water samples?
Water samples.
Water samples, okay. So just collect the water samples. If sometimes our aquatic ecosystem in the green color, and then I believe in that green color it will be a very high density of phytoplankton. But whenever it is very clean, you have to use a plankton net to collect the water samples to make it easier to identify. Because whenever we didn't have the it is very rare. It is also very boring whenever we try to identify it. Okay. So is that answer your questions, or might be you have?
Yeah. Then if this different condition, if there between a static pool like a lake or and then the river, so it will be different in the sampling process.
Basically, the methodology is similar. Even whenever we collect water samples from the oceans and the lake or river, it is similar. It is similar. Basically, it is similar because the methodology already have developed to do that. And it's again usually whenever I prefer to use phytoplankton whenever collect from the water samples. But whenever I just sightseeing and then I interesting to collect the sediment samples and then just use my water bottles and then collect the samples and then bring it to the lab. That is also if it is very rare, the phytoplankton, and then I will filter it using the plankton net in the laboratory. That is usual. Okay.
I can add a little there. Your research monitoring strategy will depend on the questions you're asking, mostly. However, you can either use random testing for statistical purposes, for statistical power, or in both lakes and rivers, you can do transect surveys. So you can go from shallow to deep part of a lake to see whether the changes are happening across the whole lake. And then down a river, you can sample from the top tributary streams down to the main river. Quite often with rivers, it's a matter of sampling where you can get access. So often people use bridges as places to go. But in Europe, of course, they use salt and there are pollution that comes off roads. So it's, if you're sampling from a bridge, it's always good to sample above the bridge rather than below the bridge. So you're not measuring the impact of pollution coming off the bridge itself.
Okay, so it's important for the point of the bridge. Okay, thank you, Mr. Pieter and Prof. Nani.
Yeah, you're welcome.
Okay, so I think it's enough. Yeah. So to end our session, I'd like to say thank you very much for our keynote speaker, both for Prof. Pieter Gell and Prof. Nanning, for very informative lecture today, and also we already has a fruitful discussions, yeah, for our first lecture series. Of course, also I apologize if in this event there is some there is some obstacle on others, but I think it is a very great event today. So I will end our session. Thank you very much, and then I will call for the Master of Ceremony, Levina. Maybe you can proceed to the certificate, yeah, or might be who will give the certificate. Might be, is there any Prof. Sabto still here, or Bu Darmanti? If not, might be I will give the certificate as for our keynote speaker as on behalf of Department of Biology. So first for Prof. Pieter Gell, this is the only we can give our appreciate for you the certificate as a keynote speaker for our international thematic lecture series. Yeah. Thank you, Prof. Pieter. Okay.
Terima kasih, Ruli.
Okay. And then for the second speaker, thank you very much, Prof. Retnanningsih, for your active participation as always. So here, the certificate for
Thank you,
Department of Biology. Thank you very much.
Thank you.
Okay, then I will end our session with Alhamdulillah and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Okay, Levina. Please.
Okay, thank you very much. Thank you very much, Mr. Wirahadian, and also thank you very much for Mr. Pieter Gell and also Professor Tri Retnanningsih Suprabhawati as to be that already give us so many informations today. It is really interesting. Also, thank you for the participants for making this previous session became more interesting. So we are going to proceed to the next session, which is the last session of the day, the closing session. Here, I would like to say thank you very much for everyone's participation today, and I would like to deeply apologize if we spoke something that caused you to feel offended, or even deeds that or any acts that cause you to feel discomfort. We are terribly sorry for that, and I really hope that everyone taking the required notes and gain so many informations from the previous session, the lecture series that held today, and I hope that it can be useful for the next future, for your researchers, or even to motivate you better to learn deeper about biomonitoring. And so I hope that we could meet again in the next lecture series. So I am hoping for everyone having a really good days ahead. And that will be the mark end of our lecture series today. Thank you very much once again for everyone's participation today. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Prof. Pieter. Thank you, Bu Naning. Thank you, Pak Ruli. Thank you, Legina.
Sami-sami.
Sama-sama.